Hello everyone, welcome to my channel. So today we will be discussing about cancer. Cancer is a very deadly disease. So a cancer once it holds you for over and over the time. So in this video we will be learning about what is cancer. We will also be talking to you about the difference between normal cell and the cancerous cells, the different types of tumor that we have, what is the cause and treatment of cancer. So this is what we will be discussing with you in today's video. What is cancer? Cancer is basically an uncontrolled abnormal growth of cells. So the growth of cells will continue. So this word, from where does it come? So according to ancient Greek, they called it as karkinos. What is karkinos? It means crab. So what does crabs do? Crabs will hold you and will not let you go. Similarly, cancer, it will hold you and will not let you go. So it's because cancer cell seems to grab on and won't let go. So that is the thing. Now, if we talk about in humans, cell differentiation and proliferation, they are highly manipulated and regularized by cell death mechanism. So there is a cell death mechanism which will go ahead and a cell division mechanism is there. So in humans, whenever the cell grows, cell differentiates, it proliferates, and then there is a cell death. So there is a cell division mechanism, a cell cycle is being followed. In healthy organisms, during this process, what happens is if one cell comes in contact with the other cell, then the replication will stop. The replication will cease. This is what I call it as a contact inhibition. Once it comes in contact, the two cells comes in contact with each other, it will stop replicating. So as a result of contact inhibition, it becomes very powerful in the anti-cancer mechanism. But what happens in the cancer cells? Cancer cells, they have lost this criteria or the characteristics of contact inhibition. Hence, what will happen? The cells will continue to divide. So once the cells will continue to divide, they will form tumors. There will be small swellings and they will start having tumors. This is except in the case of blood cancers. Blood cancers, you will not have tumors. Now, if I have to differentiate between the normal cell and cancerous cells, it's very important. Based on morphology, normal cells have uniform shapes and sizes, whereas cancerous cells, they have large variety of shapes and sizes. The nucleuses have irregular structure and have relatively small cytoplasm. So this is about cancerous cells. When I talk about normal cells, it's very normal, like uniform shape, how a epithelial cell would look like, how a blood cell will look like. It is looking like that. But whereas if I'm talking about cancerous cells, it will be varying in shapes and sizes, and it will have a very small cytoplasm. Okay. So now reproduction and cell death in normal cells stop dividing when too much of its kinds are present because of contact inhibition. These cells grow and divide in controlled manner and follow a predictable life cycle. I know once the cell starts, when it will originate and when the cell death will take place. Normal cells undergo the process of apoptosis. Apoptosis means self-destruction if they detect abnormality and damage in their organs. So if they find that this cell is not correct, they will kill the same cell. So that is apoptosis. Now, cancerous cells, they don't stop growing, resulting in the appearance of tumor, which is a cluster of mutant cells that is happening. As far as communication, so cells, they communicate with each other. Normal cells, they communicate with each other for proper functioning, whereas in cancerous cells, what happens? They do not communicate with each other. I grow. Okay. Adhesion and invasion. Normal cells, they have an external membranes that allow them to bond with the other cells. There is a bonding which is happening. But whereas in cancerous cells, what happens? Cancer cells lose the molecules that keep the cells bonded together. These cells have the ability to invade or spread to other parts of the body by traveling through bloodstreams or lymphatic systems or metastasis. That is what I call it. Next, coming and talking to you about specialization. Normal cells, they start out as immature cells and mature with a certain specialized function. Whereas what happens in cancerous cells? These cells do not mature only. They undergo apoptosis. Instead of these cells become immature over time, cancer cells are primitive and they do not have any specialized function. So there is no specialized function. We have studied about cells, their specialized functions, they form a tissue and all those things. The cancerous cells do not have a specialized function. Signal recognition. Normal cells, they recognize the signals which is there. They know when there are enough new cells and they should stop dividing. But what happens in cancerous cells? They did not recognize signals. Hence, these cells erratically reproduce mutated cells. So cancerous cells are apne marzi ka malik. So just they do whatever they think they should do. If I want to divide, the cell division will take place. There is no specific function, no signals, nothing, can rule over them. So it is completely there. They are apne marzi ka malik. Okay. So next we have, so when I talk about tumors, I already told you this cancerous cells, they form small tumors. They form various tumors. Now these tumors can be of two types. One is benign tumor and one is malignant tumor. What is benign tumor? Benign tumor are those tumors which are not cancerous and won't spread. But malignant tumor are those tumors which are cancerous and it will spread from one organ to another. All right. Now, just to give you a difference between malignant tumor and benign tumor. In benign tumor, it remains confined to the affected organ, whereas malignant tumor will spread to other organs of the body. Benign tumor, the rate of growth is usually very slow. It will grow very slowly. Whereas malignant tumor, the rate of growth is very rapid. Benign tumor, there is no latent stage, but there is a latent stage in case of malignant tumor. Benign tumor causes limited damage to the body, whereas malignant tumor, the cells will migrate to the other sides of the body. Benign tumor, there is no metastasis, whereas malignant tumor, there is metastasis. Again, what is metastasis? Cells will go and invade the other cells, will grow with the other cells. Benign tumor is non-cancerous, whereas malignant tumor is cancerous. So transformation of normal cells into cancerous neoplastic cells may be induced by physical, chemical, or biological methods, and those which are affecting or causing the cancer, they are known as carcinogens. They are known as carcinogens. Now, what are the various carcinogens? I can have a physical, biological, as well as a chemical carcinogen. For physical, I might have UV, biological, various bacteria, virus, oncogenes are there, and chemicals, arsenic, alcohol, all those things are chemical carcinogens. So, coming to what are the various causes of cancer. So, first cause I told you is physical, which is UV and ionizing radiation. So, ionization radiation like X-rays and gamma rays and non-ionization radiation like UV, they cause a DNA damage leading to the neoplastic transformation. Next, we have the chemical methods. So what in the chemical, what we have? The chemical carcinogens present in the tobacco smoke have been identified as a major cause of lung cancer. Again, alcohols can cause gastric cancers and all. Next, we have the biological causes. Okay, so biological causes would mean life. So cancer-causing viruses, they are known as oncogenic viruses and have genes that is called viral oncogenes. Furthermore, several genes called cellular oncogenes or c-onc or proto-oncogenes have been identified in normal cells, which when activated under certain condition could lead to oncogenic transformation of the cell. Now, there are different techniques how the cancer can be detected. So, the detection and diagnosis of cancer is very important before it spreads to other parts of the body. Identification of cancer genes is pivotal to prevent cancer. Now, how can we detect? Detection can be by biopsy or histopathological studies. It can be blood and bone marrow test to see the cell count that is there in case of leukemias. Radiography techniques like uses of X-ray, computed or CT, MRIs, that is magnetic resonance imaging, or molecular biology techniques can be used for identifying cancer or detecting cancer. So in biopsy, what happens? In biopsy, a piece of the suspected tissue is cut into a thin section and is examined under the microscope. It's stained and examined under the microscope to find that the particular area is infected, it is cancerous or not. Okay, this detection of cancer is there. Next, if I talk about CT, so computed tomography. CT is computed tomography. They uses X-ray to generate three-dimensional images of the internal organ. If I talk about MRI, it has a strong magnetic field and non-ionization radiation to accurately detect the pathological and physiological changes in the living tissues. So the changes are being taken into consideration. Next, if I talk about biological matters, okay, if I talk about here, I can have antibodies against cancer specific antigens. So now there are monoclonal antibodies which will combine with this cancerous cells, the antigen protein on the cell, which causes the immune response. Once the monoclonal antibody will attack, then what will happen? The immune system uh, to attack the cancer cells will be there, and hence I can always detect that if the monoclonal antibody, which is maybe radio labeled, if it comes and binds, I will know that the cells is cancerous. If they don't bind, it's not cancerous. So monoclonal antibodies are designed in such a manner. Okay, now I can have other techniques. So to detect, I can have other techniques. Other techniques are like to detect in individuals with inherited susceptibility to certain cancers. So in this case, we always can have that we may have the cancerous cells. We can take the DNA, RNA, protein, or metabolites are studied. So DNA is studied by FISH technique. Okay, so it's fluorescent immunosorbent, it is there, and RNA by microarray, proteins by proteomic profiles, and metabolites by bottomic profiles are being created, so that we have this. There are biomarkers, there are various assays, and thus various screening techniques are being done, and we can find out whether the cells are cancerous or not. Okay. So identification of such genes which predispose an individual to certain cancers may be helpful in prevention of cancers. Such individuals may be advised to avoid exposure to particular carcinogens to which they are susceptible. So like tobacco smoke, there's a lung cancer. So I can always tell them to stop taking in tobacco. Now, there are treatments of cancer. So it's not that cancer, there's no treatment. There are many treatments of cancer which is there. Okay, if the cancer, there are various stages of cancer also. So if the cancer is detected at a very early stage, then it can be reverted back. The common approaches for the treatment of cancer are surgery, radiation therapy, and immunotherapy. Against that, I have got bone marrow transplantation, chemotherapy, targeted therapy. radiation therapy. So there are various therapies again. In radiotherapy, what happens? In radiotherapy, the tumor cells are irradiated lethally, taking proper care of the normal cells surrounding the tumor mass. Several chemotherapeutic drugs are there which kills the cancerous cells, and some are very, very specific to a particular tumor. Majority of the drugs have side effects like hair loss, anemia. So you would have seen that if they're taking chemotherapeutic drugs, they have a hair loss. Most cancers are treated by combination of surgery, radiotherapy, and chemotherapy. Not an individual treatment, but a combination. Tumor cells have been shown to avoid detection and destruction by immune system. Therefore, the patient are given substances called as biological response modifiers, such as alpha interferons. We have alpha interferons, okay, which activates the immune system and helps in destroying the tumor. So we have a very well-known person who is a cancer fighter, Yuvraj Singh. He states that cancer does not mean death. We should not be afraid of it. We should go for the treatment as soon as it is detected. So early detection can always go ahead and treat cancer. Stating this, I say thank you. My next video will be on drug and drug abuse.